Hello, welcome back. Today we are going to go into a little bit of detail about DNA sequencing, particularly Sanger sequencing and NGS. So a little bit of a bigger topic, but I'm sure we can get through it. Let's start with Sanger sequencing. This was developed in 1977 by Frederick Sanger, and it was the gold standard for nearly 4 decades. It remains an important tool in diagnostic genomics when precise, high-quality sequence data is required for a defined genomic region. The key ingredient is ddNTPs. Normal DNA building blocks, dNTPs, let the polymerase keep extending the chain, whereas dideoxy versions, ddNTPs, are missing one critical chemical in the group, so the moment it gets incorporated, the chain stops. Okay? This is important because in Sanger sequencing, we don't want to just keep doing well, we can't do the entire DNA sequence. We're doing each individual nucleotide at once, okay? Each ddNTP has a different color dye attached. DDCTP is blue. I mean, you can see on the picture, I won't. DDCTP is blue, DDATP is green, and so on and so forth, right? GATTCAGC. And you can see here the peaks are representing those individual bases. So just in case you didn't quite catch the method used, as a reminder, DNA polymerase always builds in the 5 to 3' direction, adding nucleotides one by one to the 3' end of the growing chain. When a ddNTP gets incorporated, there's no 3' end or H group available to attach the next nucleotide to, right? So that's how the chain stops and it can't be extended any further. And that's how we get each individual base in Sanger sequencing. What happens in the reaction is you have millions of copies of your template DNA all being extended simultaneously. Because normal dNTPs and ddNTPs are mixed together, each copy terminates randomly at a different position, wherever a ddNTP happens to get incorporated first. So you end up with a huge collection of fragments of every possible length, and each one is ending in a colored dye that identifies the last base added. Okay. And then the electrogram here comes later. These fragments get separated into capillary electrophoresis, with the smallest first, just like gel. As each fragment passes the laser detector, it fluoresces its color, and the result is a series of peaks, as you can see, each one representing one position in the sequence with the color telling you which base is there. So for example, in this peak, you know it's an A, this one, it's a T, and so on. Reading left to right, reading the sequence from shortest fragment to longest, and reading the sequence base by base. That's how you get GAT, AAAT at the bottom. To prepare for Sanger sequencing, we really need a good amount of DNA fragments for reliable detection of sequence data. And the way that we can approach achieving this is in two ways. We can either clone in bacterial vectors, where we clone the DNA fragments into the bacterial vectors where they can be propagated and amplified within bacterial cells. If you're familiar with bacterial reproduction, you understand how that can be a good way to increase the quantity of DNA fragments that we have. And this allows for the production of large amounts of identical DNA, which can then be extracted and used for Sanger sequencing. Alternatively, probably a more widely used approach is PCR, and this is more widely used because it enables targeted sequencing of the defined DNA segments without the need for cloning. Now, NGS has superseded this. Next generation sequencing is what it stands for. It's also sometimes called massively parallel sequencing. It means the same thing. We'll go through the purpose, advantages, platforms, terminology, bioinformatics, which is a critical component of NGS. And we'll also go through choosing how much to sequence because the data that you can produce is never ending and it's not convenient to sequence everybody's entire genome. There'll be a large amount of data for sometimes only wanting to find out whether or not they're positive for like a BRCA gene for cancer. But we'll get to all of that in the end. Let's start off with the purpose. NGS refers to a suite of high throughput sequencing technologies which emerged around the mid 2000s and have transformed genomic research and diagnostics. Unlike Sanger, which processes one fragment at a time, NGS enables simultaneous sequencing of millions of DNA fragments in parallel. The advantages are that it allows for rapid, large-scale analysis of genetic material, greatly increasing the sequencing speed, so the turnaround time at a lab, and also the data output while significantly reducing cost per base. There are multiple platforms that do this. Multiple are currently in use. They can be broadly classified based on whether or not they incorporate PCR amplification as a step during library preparation or sequencing, or whether they use a PCR-free approach to preserve sequence fidelity and reduce bias based on which one you use. Each platform has strengths and limitations, and these are related to read length, error rate, throughput capacity, and compatibility with different sample types. Clinicians and researchers that are doing NGS, they have to critically evaluate each platform's suitability in the context of their specific clinical or research objectives and goals. Terminology that we need to be familiar with in order to understand NGS is as follows. A read refers to the output of an NGS sequencing reaction. So it's a single uninterrupted series of nucleotides that represent the sequence of the template. Read length is the length of each sequencing read, and this is always represented as an average read since individual reads have varying lengths. Read depth refers to the number of reads that include a given nucleotide in the restructured sequence. Due to error-prone sequencing reactions, random errors can occur, right? So therefore, about 30 times is typically required to ensure each nucleotide sequence is accurate, but this will vary depending on the circumstances. When we say library, a library is a collection of randomly sized DNA fragments that represent the sample input. Different NGS technologies generate libraries in different ways. And then alignment. Alignment is mapping a sequence read to a known reference genome, and that's the last terminology. The reference genome is the genome that we compare the patient's sample to when we do NGS. Bioinformatics, let's talk about both, I'll go into detail about both pipeline and variant creation and interpretation. So in NGS, short DNA reads are first aligned to a reference genome, as mentioned earlier, and then bioinformatics tools will compare these aligned reads to identify consistent differences, such as a single nucleotide variant, while at the same time filtering out sequencing errors. So this includes the design of algorithms, software tools, and databases that facilitate the integration of statistical, mathematical, and computational approaches and pairing them with molecular biology and genetics. So as you can imagine, it's a little bit much for the human brain. We need the assistance of bioinformatics tools. So the typical sequencing pipeline involves three steps. The first one is sequence alignment. When we do this, raw sequencing reads are mapped to a reference genome, and this process, also called read alignment, relies on sophisticated algorithms to account for sequencing errors, repetitive regions, and alignment ambiguity. As you can imagine, there's lots because there's lots of sequencing data. Once aligned, the next step, step two, is variant calling and annotation, and this is where we identify regions in the genome that differ from the reference sequence. These might be CNVs, insertions, deletions, and structural variants, but detected variants are then annotated with biological information such as their location within the genes, the predicted impact on protein function, and known associations with the disease. The third step then is variant filtering and prioritization. As you can imagine, you'd probably get a lot of variants and you want to prioritize which ones are clinically relevant. Given the large number of variants that are typically identified in the genome, further filtering is necessary to distinguish pathogenic variants from benign variations. This step involves integrating data from population bases, gene disease associations, your knowledge of inheritance patterns, and computational predictions to prioritize high confidence variants for further investigation and clinical interpretation. When we do this classification, there's a scheme here, and it ranges from pathogenic to benign. It involves a thorough evaluation of each variant using a combination of bioinformatics tools curated databases, scientific literature, and clinical information to classify the variant's individual potential impact on health. Although the classification scheme is great and helpful, variant creation and interpretation goes beyond assigning a categorical label because it requires the interpretation of the variant within the clinical context of the patient. So even if it's, pathogenic or benign, we also have to consider the phenotype, their family history, the inheritance patterns, and other clinical data that we have. Finally, we want to choose how much to sequence. So there are several approaches for choosing how much DNA to sequence when looking at pathogenic SNVs, and this typically depends on our goal, right? So if we want to do targeted gene sequencing panels, this is usually chosen when only DNA sequences of selected genes are targeted and captured in the library, and the panel will consist of genes known to be associated with specific diseases or phenotypes which we predict that the patient may have. So these can be cardiac panels, cancer panels, nephrology panels, pediatric panels, based on what we're trying to figure out. And by doing this, it reduces if we want to try and avoid incidental findings and just confirm something we're probably already um, estimating based on things like family history or phenotype or other clinical information. Oops. We can also do whole exome sequencing, where all the DNA sequences that code for proteins or the exome are targeted and captured prior to sequencing. It's good to know that the exome accounts for about 1 to 2% of the genome. So sometimes this isn't enough to give us a conclusion on what we're after to help the patient. So another approach is the entire genome. This is where all the DNA sequences in the genome are sequenced. Although there are regions, because it's so big, there are regions that might not have full coverage and might be a bit difficult to sequence. These can be like areas with high GC content or highly repetitive regions. So there are pros and cons. With with option four, we can do a targeted bioinformatics analysis. You can think of this as like a two-stage filter. So stage one would be wet lab, and this is where you choose how much of the genome to physically sequence. So do you want to do a targeted panel, the whole exome, or the whole genome? And then we do bioinformatics to narrow it down further. So even after sequencing broadly, you don't have to analyze all of it. You can computationally restrict your attention to the specific genes of interest, essentially ignoring the rest for now. And the key insight is that the raw data is still stored. So if your targeted bioinformatics analysis doesn't find an answer, you can go back and interrogate other regions without resequencing the patient. You're not throwing the data away, you're just deprioritizing it. This is particularly useful for reducing variants of uncertain significance to clutter your report. Broader sequencing captures more, but you only then surface what's clinically relevant at the moment. For commercial labs, usually targeted gene sequencing panels are used because you just want to confirm what is being hypothesized, essentially. And for the final slide, I've got a nice summary that I took from Macrogen to help you distinguish between Sanger and next generation sequencing. Hope this helped, and yeah, see you later.